Hello,
Voor altijd.
everyone.
Je hebt dan toch de goede bijgehouden.
Can everybody make sure that they are muted? I want to start this new participant. Okay, so hello everyone. My name is Jan van den Holland, working as a business development manager at GreenSoil. Thank you for joining us today to discuss the outcomes of the investigation that GreenSoil and Wageningen University carried out together on the complete and simultaneous dechlorination of chloroethenes and chloroform at high concentrations. This webinar is given by John Dijk, GreenSoil manager, GreenSoil R&D manager, and Tom Bosma. He's a researcher at Wageningen University. John started his career as an academic researcher. He got a PhD degree in microbiology and worked as a postdoctoral researcher. In 2007, he became R&D manager for a remediation contractor, where he translated his knowledge into practical solutions and developed new remediation techniques. From March 2016, John joined GreenSoil as R&D manager. Within GreenSoil, his main tasks are developing new remediation techniques, investigating the potential of bioremediation of emerging contaminants, and finding cooperation with universities and knowledge institutes. He designs and conducts laboratory and pilot field tests to investigate the feasibility of bioremediation techniques by following up the processes of bioremediation. Tom graduated at Wageningen University as an environmental microbiologist. After his PhD about biotransformation of subsurface pollutants, he moved to the Swiss Federal Institute of Environmental Sciences in Switzerland, where he was involved in ecotoxicological research and an initiative for a research project to assess the environmental and social impact of hydropower. From 1996, he worked at TNO, a Dutch research institution, and Utrecht University, where he dwelled around in various management roles. He is active since 2015 at Wageningen University, where he is involved in biodegradation research and education. This webinar will last about 45 minutes. Afterwards, there will be some time left for questions. Feel free to write your questions in the chat box or send us an email. I will pick them up during the presentation and address them to John and Tom. Should you have any other questions that come up afterwards, just send us an email and we'll come back to you with the answer. I would like to point out that this webinar is being recorded. The recording will be made available by email to all registered users. May I also ask you to turn off your microphone and camera and enjoy the webinar. Before handing over to Tom and John, I also would quickly like to introduce our company GreenSoil. For those that are not familiar, we are an international and innovative general contractor specialized in biological and sustainable ground and groundwater remediation. With a team of about 25 dedicated people and through the implementation of innovative techniques, we perform remediations for a wide range of clients, and we have successfully delivered projects all over the world. We put a strong emphasis on incorporating biological techniques and client-specific needs to achieve the set-out targets and have an extensive track record of beautiful projects that we delivered in the past 5 years. As I don't have anything further to add, I'm going to hand over to John and Tom, and I'll come back to you at the end of this webinar. John, please go ahead.
Yeah, thank you, Jan, for the introduction. Switch off the pointer. As Jan already mentioned, basically will be a dual presentation, so I'll start with some results from the ongoing Tarragona project, with a bit kind of a focus on the results that are also relevant for the second part, where Tom will basically tell about the thesis research that was done for this project, where we specifically investigated the influence of chloroform on the complete dechlorination of chlorinated ethenes. So a little bit of site background. It's an industrial site in the harbor of Tarragona in Spain, which is heavily impacted with chlorinated ethenes, of which PCE is the parent compound, and chloroform. We do the project together with Tau as a consultant, who is also doing the monitoring. And maybe an interesting thing to know is that the average groundwater temperature here is 21 degrees Celsius. For us in Belgium and the Netherlands, there's of course a quite high temperature, but very positive for microbial growth, as we will see during the presentation. Also some challenging conditions. On one hand, we have, due to, of course, the influence of the sea, a very brackish salt groundwater with a high electrical conductivity, high sulfate concentrations. And on the other hand, of course, there's the possible toxicity of chloroform. If you look at the scientific literature, for example, it's known that chloroform, starting from concentrations of 5,000 micrograms per liter, can already inhibit the growth of Dehalogenation. And as probably most of you know, this gene is in general, of course, very important for especially the last two steps of the dechlorination of ethene. So basically the conversion of cis and vinyl chloride to ethene. If you look at the concentrations here at the site, so I indicated some maximum concentrations just to give you an idea about the heights of the different contaminants. You can see that for PCE, it's already quite close to the maximum solubility of PCE. There was already quite some vinyl chloride present at the site before we started, of course, indicating that the process of dechlorination was already ongoing. And as you can see, very high concentrations of chloroform, far above this 5,000 micrograms per liter that we found in literature. Some little bit of history of the remediation itself. Basically, it was started with a laboratory degradation test in 2014, followed up by a field test in 2015, and then a full-scale remediation started in 2017. The full-scale remediation basically consisted of circulation of groundwater and semi-continuous dosage of electron donor. So basically, we just try to circulate the groundwater at electron donor and kind of create an in situ bioreactor. And this was combined with DNAPL extraction and removal. You can see that a little bit here in this schematic drawing. So here we have the groundwater matrix. So basically you extract water from here, from this well, this extraction well, from 3 to 9 meters below ground level. The water goes, well, basically through the container. There's not much here, but the only thing is that we have this DNAPL removal device in line, and we add our electron donor, and then the water is with a closed system infiltrated again here. So basically you create a biological active zone here where the dechlorination is being stimulated. Here you can see a top view of the site. So you can see that there are basically 3 different areas. So we have basically 2 kind of outer areas, one here, one here. And this is what we consider basically the source zone. The active remediation system is basically divided in 2 different systems, mainly because of the concentrations. So we keep them separated. So we have the system 1 and system 2, but I won't go into detail too much in this presentation. Just to give you an indication for the DNAPL removal, you can see the graph here of basically the amount of cumulative amount of DNAPL being removed. As you can see, it's a very low quantity, but if you look at the basically the curve itself, you can see that at least the extractable amount of DNAPL that was in the water was being removed. A little kind of schematic representation of the progress of the remediation throughout the years. So this is in December 2016, basically before we started the full-scale remediation. With these colors kind of indicated the percentage of solubility of, in this case, PCE. So the pictures are only for PCE, which was kind of the main target. So the darker the color or more purple red is of course higher concentration, close to 100% solubility. And towards the green are the lower concentrations. So just to give you an idea, so you can see here it's different colors. It's more, of course, more the outside area, and here more the source zone. And you can see there are also some places like here is a loading dock. Here there are tank pits where we couldn't install system, where there might still be some contamination present below. So that's why the question mark. So there can also be some influence from here towards our basically our remediation area, which makes it a bit more challenging. 2017, so the first year of the remediation, you can already see that there's some changes, especially here there are of course a little bit higher concentration, but especially here in 2018, you can see that the concentration kind of went up, basically also due to mobilization because of the circulation of the groundwater. But also we switched the wells, so you can see the wells here below. So the small numbered circles are basically the infiltration and extraction wells, and we changed the configuration, so which one are infiltration and which one are extraction, in order to reach all the areas. Of course, based on the monitoring results, sometimes you see some areas where they kind of concentrate the problem or the area you need to remediate. So sometimes by changing this configuration, you can have a better outcome. But it also means that sometimes it fluctuates a little bit depending on the actual configuration. And then in this year, you can see that basically the outside areas are relatively clean. So you can see by indicating by the green color. And the higher concentration, they are kind of concentrated in this area where also is basically the sewer line. So basically where we think that the contamination enters the groundwater matrix. So a strong concentration in this area. Because it wasn't really known how much contamination was present at the beginning, also due to the fact that not all the parts could be reached with the investigation. It's of course difficult to have a kind of progress by means of mass reduction. Of course, you can look at the groundwater, but yeah, I already mentioned there's DNAPL present, probably also a little bit of soil contamination. So we did another approach, basically to have at least some kind of feeling for the total mass reduction, and therefore also the progress of the remediation, because it, as I just mentioned, in some areas we still saw high concentration, depending. We said, well, at the same time we saw that the complete dechlorination was going on. So of course, if there's still a lot of DNAPL present, for example, then you also remove a lot. So just in order to have a feeling for the amount, we try to kind of make an estimation based on different values. We started with more like literature values, so typical degradation rate also for specific microorganisms, since we knew the amount of microorganisms present, which I will show one of the next sheets. We also did some basically degradation tests ourselves on both on the site and in the lab to have kind of an idea about the actual degradation rate with material from the site. And then of course we have the lab tests done by the Wageningen University that will Tom will also show. So you see there's a big range in the total amount of PCE being removed due to degradation. Of course you could say that the lab tests give probably more an overestimation because there are ideal conditions. This one more slightly probably a more underestimation. As you can see, at least the order of magnitude is in a few tons of PCE being removed due to the degradation. If you then look to the concentrations a little bit more into more detail, so I selected a few wells indicated here by the blue circles. So 4 different wells, 2 more in the source zone and 2 in the other system. So I'll start with this one. So we have the concentrations here. These are in thousands, of course, with the Dutch and English. There's sometimes a bit confusing with points and commas, but this is 300,000 micrograms per liter. And on this axis is basically the ethene concentration indicated here by the blue line. And these are also so this is 14,000 micrograms per liter of ethene. Then here with the bars, we have the different chlorinated compounds. So we have the dichloromethane, chloroform, PCTC, cis, and vinyl chloride, of course. And then in time. So see for this well, First, as I mentioned, we have a little bit of mobilization due to the circulation of groundwater. You can see that you basically start with, in this case, with chloroform, TCE, and also here some PCE. Already the degradation products are present, at least to some extent, basically the cis dichloroethene. But you can see that already here it starts to the amount of cis DCE, in this case the relative amount, increases. Also here the phenyl chloride, and then you can see a clear decrease in concentration in time. It kind of stabilizes around a relatively lower concentration, but you can also see that there's still some parent compound, especially the chloroform, is still present. And you can see a clear increase here in ethene concentration that stay relatively high, up to tens of thousands micrograms per liter of ethene. If you look at this well, another one with more farther away from the source zone, you can see that here the ratios are a little bit different, relatively more, well, a lower concentration, of course, if you compare with these, but the ratio, there's a little bit more chloroform here, also TCE and the cis dichloroethene. You can see that especially the amount of cis DCE increases. Also here you can clearly see the dichloromethane being produced from the chloroform. And then also here a clear decreasing trend, with here still a little bit of a phenyl chloride. And also again the increase here in ethene up to, at least in our experience, very high ethene concentrations. You can see here 40,000 micrograms per liter. This is one of the highest we ever found on a project. Probably also had to do with, as I mentioned in the beginning, the relatively high groundwater temperatures. Then another one more located in the source zone, but a little bit more to the other side. A bit of similar trends. So here we started. Here is more PCE present in the beginning, already the degradation products. And then also here you see a clear decreasing trend in concentration and also increasing trend in ethene concentrations. And then in this one is basically really the source zone. So you can see a lot of chloroform here. Also the PCE and dichloromethane. And you can see that here there's no, if you look to the ratios, doesn't change that much. Throughout the time, there's still high concentrations, a high amount relatively of chloroform and PCE. Of course, there is fluctuation in the total.
John, sorry, I lost your presentation.
Is it? You see now the full screen again?
Yeah.
Yes.
So one back. So we were here. So there is fluctuation in basically in the total concentrations, but as already mentioned, that there's also had to do with basically the switching configuration of infiltration and extraction. But still, you can see that also the concentrations here remain relatively high because it really is in the source zone. We also looked basically at the microbial population in the different wells. So basically the focus on the dechlorinating microorganisms. Again, here we indicated the, okay, now I lost the red dots. So here we located the different wells, and here the results. So we looked, here you can see the total bacteria. We looked at the Dehalococoides here and Dehalogenimonas, which are basically more involved in the dechlorination of especially cis dichloroethane and phenyl chloride to ethene. We have the Dehalobacter, which can be involved both in the degradation of chloroform and PCETC. The Geobacter, of which is known, it can also be active in the near presence of DNAPL, so really high concentrations of chlorinated ethenes. Then we have the two phenyl chloride reductases, VCR and BVC. And then here we have the chloroform reductase. The orange line basically represents kind of threshold level, for example, indicated by the EPA and some other parties, which is basically is for Dehalococoides. So this is this 1 to 2 to the 7th cells per liter, which is basically considered as a kind of a threshold. So if you have higher numbers, you can basically assume that complete dechlorination is going on. Of course, we also have the ethene concentration to confirm that, but just as an extra check, it's also not good to see that in general these numbers are way above this threshold. So what you can see here is that already before the remediation, we had really high numbers, basically for all the targets that we tested. Only for this one, we didn't test it the 2-time because it's a relatively new target. So we tested that one only the last in 2019. But you can see already high numbers present at the start of the remediation, and also increasing trend for basically all the targets in this well. For this one, where we really where the concentration of chlorinated compounds were higher, you can see that the starting numbers were significantly lower if you compare to this well. But also here you see an increasing trend during the remediation. And for this one, basically the same here. Yet the starting point was also higher, with higher numbers. Also, especially in 2018, increase. And here you see a little bit a decrease in 2019, which also has to do that if you go back to the sheet. For W25, you can see that basically most of the chlorinated compounds are already gone. So that's not much to do for the dechlorinating microorganisms. If you look at the microbial community, so the complete community, this is of course only for one well where we did next generation sequence. But also here you can see that the Dehalococoides is a big portion of the total microbial community. In this case, it was 27%, but also based on the qPCR, on average, about 20% of the total bacteria consisted of Dehalococoides. This is, I think, something just to remember when Tom will show his results. Of course, in the beginning, I mentioned that the presence of high concentrations of chloroform are known to be inhibiting for Dehalococoides. Well, basically here we see that Dehalococoides is really high in numbers in most of the wells. So we try to get a little bit more understanding, also the relation in this case between ethene and the chloroform concentration. So here we took several wells. Some of you are already demonstrated before, but here you can see basically in green the ethene concentrations in those wells. So the concentration is on this side for ethene, and then the brown bars are the chloroform concentration in those wells, which is on this side. You can see there's not a clear correlation, but I think the take-home message is that here the orange line is basically the 5,000 micrograms per liter, so the concentration mentioned that could inhibit Dehalococoides. You see that the chloroform that we find in the different wells, it's much higher, up to 140,000 micrograms per liter. But in all those wells, we find also ethene in relatively high concentrations. This is of course a very interesting finding, and then I come basically back to the conclusions. So we could basically demonstrate complete dechlorination in a brackish environment despite very high concentrations of chloroform. We also saw that chloroform was basically degraded to some extent, at least, to dichloromethane. But we couldn't detect any chloromethane. It was measured, but it was always below the detection limits. We could also demonstrate basically that Dehalococoides was a very dominant group, so to say, a very relevant group in basically all the wells with a high number. And that's basically also the connection to the results that Tom will now present. So basically the connection on the fields, we see these high Dehalococoides numbers. But what is really going on with the chloroform? So that's one of the reasons why we decided, together with the world, to look into it into more detail in the lab by doing different degradation tests. But that's something Tom will show. So Tom, the floor is yours.
Yeah, thanks. Thank you, John. I'll wait a while until your presentation has disappeared.
I can stop sharing, maybe
Exactly.
it helps if
Yeah, otherwise I cannot share. Here we go. Okay, so thanks again for inviting me to also present our results about this very interesting project, actually. We did the work at Word together with a master student who was assigned with the task. I think maybe some of you recognize the picture on the left-hand side, which is the site at Tarragona, and on the right-hand side, you see the laboratory incubations as we had them in the lab finally. What I would like to discuss is first a small presentation about the work that we do at the Laboratory of Microbiology relating to pollutant research, show the lab results, of course, and then draw some conclusions and enter into a discussion. I had also a small bonus or a sidestep, and I would like to show something about people's toxicity towards dechlorination. Yeah, at the lab at microbiology, there is actually a very long history of dehalogenation research. I was involved myself with the start of this. I did a lot of studies about dechlorination, mainly of chlorinated pollutants, of course, chlorinated benzenes, also hexachlorobutadiene and PCE. What we recently find is that also in pristine environments, so which are not polluted, there are many dehalogenators present, and they also have dehalogenating activity. So that's an interesting new view actually on dehalogenation. We have been thinking for a long time that it was a newly developed property of microorganisms, but it appears to be very old actually. And the reason is probably that in marine environments you have a lot of halogenation reactions going on, and as a complement also dehalogenation reactions have been evolving there. Then at polluted sites we are mainly interested in complex interactions between pollutants. You could say that for the single compounds we kind of know what's going on, but when you mix the compounds, when you have mixed microbial populations, it gets a little bit more complicated. So we look at mixed degradation kinetics. We want to look at competition effects and also toxic effects on dechlorination pathways. Then PFAS is a new topic that we are also starting to study. The research is going on for a little bit more than a year now, but of course the pandemic complicated things a little bit in an effective startup of those. But having said that, what we are interested in from that point of view also is what is actually the impact of pollutants on microbial ecosystem services, because microbes do a lot of things for you in the environment, like nitrogen cycling, sulfur cycling, and also the breakdown of halogenated compounds and other compounds, of course. And so how does that interact? We also use modeling. I won't talk about that today. And we have a recently new development that is the facility that is called Unlock. And Unlock is a facility that has been paid by NWO, or is being paid, I should say. And it's focused on the biodiscovery of all kinds of new pathways, new microorganisms. It has a facility for modular bioreactors, and it has a lot of potential for parallel cultivation. That's a contribution done by Delft University. And there is a big data center, because when we do modern biotechnological and microbial research. We generate a lot of data. And so we also have a facility to handle those and to store those. So when you're interested, you can go to the website, look at mlog.nl and have a closer look. Then back to the project. So on the left hand side down, I also try to make this nice laser pointer. Here is the site. What we received from the site is a big pallet with groundwater samples. The samples were stored like this, so they were stored at about 4 degrees probably in these nice packs. And actually they were really cold still when they arrived at the lab from Spain. We initiated a lot of cultures. We did our analytical work on those. And in the end, we generated results about biotransformation patterns and also some microbial community analysis. Then going back to the site. So the idea actually was, because GreenSoil wanted to know what the impact of chloroform could be on the dechlorination of PCE. But as John already showed, actually the dechlorination of PCE went very well at the site, and also the degradation of the chloroform went very well. But anyway, what we did is we took samples from the highly polluted part of the site, and we received samples from 1001D. That was a well that John also discussed, which was relatively low in PCE and low in chloroform. On the opposite, we had also a well that was number 14, which was high in PCE and high in CF, in chloroform. We had a well that was low in PCE and high in CF, and then also the high PCE and low CF one. And I think number 18 and 1001D were also discussed by John. And these two wells are different from what John discussed. Well, this is again an overview of what we did. And I think it's almost a repetition of what I just said. So we did incubations in these small bottles that you saw before, with low concentrations of PCE, low concentrations of CF, with high concentrations of PCE, low concentrations of CF, high PCE, high CF, and low PCE, high CF. And we took samples from the corresponding wells to do these studies. And the idea behind that was that then you have the possibility that the populations that you have from these wells in your groundwater are already kind of adapted to the conditions that you give them. This is a table summarizing the same, but also mentioning the concentration that we then consider low and high. So for PCE, 70,000 micrograms per liter was low in this case, and for chloroform, 12,000 micrograms per liter was also low. And on the high end, we had 60,000 micrograms per liter of chloroform and 85,000 micrograms per liter for the PCE. And maybe it's good to notice when you look at this 12,000, that is more than twice as high as the concentration that affects the halococcoides, because the limit for that is 5,000 micrograms per liter. Then we also checked different e-donors in the project. We also had formate in there, but I won't show results for those. So I will only focus on the Dehalogenation. That's the electron donor that GreenSoil normally uses in the remediations. And tween 80, that's a surfactant. And the idea was that maybe tween 80 can also help to mobilize the pollutant. And we wanted to know if it would be toxic. Then I immediately go to the tween 80 and Dehalogenation results. And essentially the message in this picture is when you do an incubation with chloroform and PCE at high concentrations, and you add tween 80 or Dehalogenation, the result is not that different. Interesting is what you see with tween 80, that when you look at the chloroform, I realized that this is a little bit complicated picture, but this is the chloroform removal here. What we see in the lab very clearly is that we get a 1-to-1 dichloromethane appearance, but the dichloromethane also disappears again. And that was a reason to do a respike of the chloroform, but what you can see is that we could not finish that experiment. And the reason is that here COVID appeared. And we had a close down of our labs for, I think, more than 3 months after that period. So then actually we couldn't work on these bottles anymore further. With the Dehalogenation, but we also see the removal of the chloroform, which is a little bit slower in this comparison. I must say that we have many bottles where it also disappears immediately. The dichloromethane also appears again, and what you can see here very clearly is we did the respike of the chloroform simultaneously, but in this case this was too early, and we just get more dichloromethane when we do that, instead of a removal of the dichloromethane. But the good news is that the chloroform disappeared again immediately. But one lesson that we learned from this experiment is that if you want to give additional pollutant, you have to wait until the complete transformation pathway has been done. Then for the PCE, you see the typical pattern, it disappears. You get cis-dichloroethene, and we also see ethene finally coming up in this experiment. And that's here we see an accumulation actually of phenyl chloride. Here we also have that, but please note that here we are really at the high end of both concentrations. So chloroform is going to be very toxic for the dehalogenators of the PCE. And that is what you see back in the accumulation of the phenyl chloride. And that was very consistent in all the experiments that we have done. At the high concentrations of chloroform, phenyl chloride, dechlorination was really blocked. This is a kind of summary of all the combinations is coming into these two slides. So at low PCE, low CF, in this case, what you see is chloroform disappears. It's this light blue line here, and then over there, oh sorry, this is PCE. The color code is not consistent. PCE, oh yeah, PCE is here dechlorinated, and we get phenyl chloride as the end product. And also here you see that in this experiment we did the respike actually a little bit too early of PCE, which had as a result that the phenyl chloride was not further dechlorinated in this experiment. And here we had to lock down again, so we couldn't study these cultures further. What we see here for the chloroform, that's the second panel here, is being removed. We see a short appearance of dichloromethane, which also disappears. And then after a respike of the chloroform, we see the same effect. At high PCE concentration and low chloroform concentration, it's about the same. We see PCE disappearing and we see phenyl chloride accumulating, but what you also see that in the end, also phenyl chloride is dechlorinated. And when we do then a respike of the PCE, the phenyl chloride accumulates again and it disappears again. And I think here this red arrow is a respike of again of the chloroform of the PCE, sorry, yes. And then, well, here it's a little bit, it's just 2 data points after that one. So, but what you see also is that consistently the ethene is accumulating during all this experiment. And for the chloroform, it's kind of similar to the left panel. So I just move on. Then here we have the experiment with the high PCE concentration and the high chloroform concentration. And here you very clearly see what I initially mentioned, that when chloroform is high, phenyl chloride accumulates and is not dechlorinated. And that, well, as mentioned, we saw that in all experiments. What you also see in this experiment is that the chloroform at the high concentration, after a while, gets dechlorinated. We see production of dichloromethane. And in this case, after the respike of the chloroform, we see an accumulation of further accumulation of dichloromethane. But again, like in the previous slide, actually we did this respike maybe a little bit too early. We should have waited before the dichloromethane really went down. And the problem is that dichloromethane is almost as toxic as the chloroform. So in fact, you're doubling the high concentration then of this toxic component again. Then with the low PCE and the high chloroform, you see again here this accumulation of the vinyl chloride, which is important. And also what you see is when we do the respike of the PCE here, we see a little bit scattering, but it's dechlorinated again. And ethene in the end is coming up at a very small amount, but I'm also not surprised when here maybe there is some analytical error going on. And with the chloroform in this case, you see that it's actually not degraded, and so we shouldn't have done the respike here. Maybe it's good to give a kind of summarizing result. This is a picture that was done from the well with the high concentration of the PCE and what we call the low concentration of the chloroform, so 12,000 micrograms per liter. And what you see in this picture is that yes, we get PCE dechlorinated, and yes, we get ethene produced over this line. And when we do an additional spike of the PCE, when the VC has been removed completely, then we get a reappearance of vinyl chloride, which disappears again. And then we can again respike the PCE and see an additional dechlorination and formation and disappearance. In this is not the VC yet, I guess this is the CDC. And with the chloroform, it's similar. The chloroform here is very difficult to see due to the scaling, but it disappears. We should get, I have John on the legend, so I remove John. Here we have DCM, the gray line. So here you see it's very quickly produced actually. Then when it has disappeared, we can give a respike of the chloroform, goes up and down again. This is PCE, sorry. It's this small blue line here. It comes up, it goes slowly down. And we see then again, we should see DCM, but actually we didn't see it. And the possible explanation could be that in that case, the DCM is also quickly removed so that you don't see the intermediate product. So this is a very good representation what we have seen in many of the bottles actually. That's why I call this the summarizing result. Now I have, yeah. So this I wanted to delete, but I forgot it. Then we also looked at the microorganisms. We also had some results about the community analysis. What was striking there is that we didn't see Dehalococoides, for instance. And when you look at the pictures here, I don't think it's necessary to go to all the details, but what we see is that non-typical dechlorinated strains, but strains that can dechlorinate, are present at relatively high abundances. This is also community analysis, by the way, but we don't see the typical dechlorinators in our experiments. And sometimes at some point they come up a little bit, like here and there, but as you can see, it's a lot lower than these ones. And probably what we think is that the reason is that because we are always above the toxicity level for the chloroform in our cultures, the Dehalococoides and also the Dehalogenimonas, they don't get a chance actually to develop. And maybe we immediately even knock them off when we do our additions. And of course, an important difference between the lab experiment in the way that we did it and the field is that in the lab we mix the bottles completely, and so all organisms are exposed to the same concentrations in the end. On the other hand, we see good dechlorination reactions in the lab, so apparently the other organisms which are capable of dechlorination can take over a lot of the activity that you need to get complete dechlorination. And that's the good news, I guess. Okay, I think I already mentioned many of the conclusions, but here they are again. So PCE is completely dechlorinated to ethene, like in the field, where we also saw that in the presence of CF and also at concentrations that are known from literature to be very toxic to the typical dechlorinated organisms. PCE only dechlorinates to vinyl chloride when the concentration of chloroform is too high, in our case, to 60,000 micrograms. The presence of DCM is also toxic, so DCM must disappear in order to achieve the complete dechlorination of PCE to ethene. We have not seen toxicity of tween 80. And dehalogenation is at very low levels in the lab, and this is really in contrast to the site data. Then I just want to mention that COVID sucks. And then we had a kind of bonus question. We started our research on PFAS biodegradation, as I mentioned. So we were also interested to see how PFAS would affect dechlorination, and we used actually the cultures from this work to test that. And here you can see a result. So the tar is for the Tarragona, the 500 is a low level of PFAS, which this was based on a microgram per kilogram of soil concentration. This is what we had in the water phase. And so what you see is when PFAS is at 300 micrograms per liter, we have a rapid dechlorination of chloroform. We have a rapid disappearance of PCE. And in this case, we also added dichloroethane to the cultures. And you see that we can do a lot of repetitions to keep the dechlorination active. When we have the higher concentration of the PFAS, we cannot do that many repetitions. From the slope, it's hard to tell if it's really slower. But yeah, so we see here a first indication. We think that PFAS might be toxic actually to dechlorination. I want to explicitly say that this is not relevant to the field site that we were studying, because there we don't have this pollutant present. But I thought it might be interesting to show and also to let you know that we work on this. And of course, the other side of this experiment was that we also wanted to know if a dechlorinating culture would be able to also defluorinate PFAS. But that we didn't see. At least we don't have convincing proof yet. And with that, I would like to finish and start the discussion.
Perfect. Thanks, John and Tom. I do have some questions that came in. First off, some questions for you, John. Do you know how much PCE and CF chloroform were removed by the end of the project?
That's a good question. It's still kind of ongoing. Also, the project is ongoing, so we haven't finished yet. As I mentioned in one of the slides, we did do an estimation for at least for PCE. We haven't done for chloroform yet. It's on the list, but also for, of course, for PCE dechlorination, there's much more is known in literature, like degradation rates, kinetics, etc. But yeah, what I also mentioned, one of the complicating factors is, of course, that we don't really know what was like the starting mass. So then it's always difficult to, I mean, you can do it for groundwater concentrations, but I think that's not really relevant if you know there's a lot of DNAPL present. So yeah, I cannot give a good answer for that. So besides the estimation we made for PCE that I showed in the sheets.
Another quick question. Can you share what type of literature you used to calculate the removal rates?
So that was the first one. That's a good one. I can't recall the exact because it has been 2 years, something. I have to check it, but I know that we did it both for like the values for the reported for Dehalococcus and Geobacter. Combine those values. Have to check the exact reference.
Yep. And another question: why did we involve additional lab studies at all in view of our excellent field results? Maybe something you can share.
For me as well. Yeah, well,
Yeah.
basically, it's of course, like I mentioned, it's known from literature that, for example, the chloroform can have a inhibiting effect, as already mentioned by both me and Tom. In the field, we didn't really have clear indications that it was happening. So that was one of the reasons also to have a better understanding, and also with regards to the degradation pathway of chloroform. So the dichloromethane, we couldn't detect any chloromethane, for example. A few reasons to look further into that into the lab, because then you can better check it because it's a closed system. Of course, what Tom already mentioned. Downside is, of course, it's in the field you have more heterogeneity, so there's less chance of course that a compound is inhibiting, because always spots kind of microorganisms can hide or protect themselves from the high concentration, while in the lab it's a homogeneous system, so this way the results cannot always be compared one-on-one, but at least to gain a better insight of those processes. I think it's very important to have those kind of lab tests.
Yeah. A question
maybe Jan, if I can add to this, because I also see a question from Peter Middelder.
Yeah, just want to
connect it actually. What did I want to? Well, to the question of Peter, so he was asking if dichloromethane is further dechlorinated or not.
Yep.
And it is actually, it's removed, so it's not accumulating. And we now have cultures that seem to be able to grow with dichloromethane in combination with Dehalogenation, the electron donor. They also grow without the Dehalogenation, but that is a lot slower growth that you have then. So as an answer to that question, yes, dichloromethane is actually being removed. And in fact, what we see in the lab then is also consistent with what you see in the field.
Yeah.
Because also in the field, you don't actually really see chloromethane in high levels, but you see dichloromethane popping up and disappearing again. So that's really similar.
Yeah. Following that question, Tom, I want to close this one down. Do you know what happens with the DCM and which microorganisms might be involved in this degradation pathway?
No, actually we don't, and that's a follow-up question that we are now addressing with a new master student.
Yeah.
But still, we are working under COVID restrictions, so it's going slower than I would like to. Let's put it that way.
Yeah.
And it's more difficult because for dechlorination you have a very clear pathway and a very selective pathway from the point of view of the microbes.
Yes.
Well, probably the dichloromethane removal and biodegradation pathway is less specific, and possibly more organisms are involved in that. So it's a little bit more difficult to select for that. But the idea was, when we have a culture that really can grow on dichloromethane alone, then you can really enrich for those organisms. But that's much more difficult than adding another electron or carbon source and having the cultures grow. So that's a slow process, but we are definitely interested in that. Okay.
Then another question that came in from Charles Pels, and also a follow-up question on that. How do you explain Dehalococcus was observed in the field in high concentrations and not in the lab? Is it possibly the result of a heterogeneous distribution of the biodegradation processes in the field? And following that, How would you conclude how representative are lab tests for situations in the field? I think that has been a topic of previous webinars as well, but I also this also shows that is always a nice discussion to have. How maybe, and yeah, it could be a question for both of you. So I'll leave it up to you to decide who responds to this one. But I think it's an interesting subject.
Maybe you can do a double answer.
Yeah.
I can start with a short.
You start, John. Yeah.
Yeah. Okay. Yeah, I already mentioned, like, I think the heterogeneity is a very important thing that has been so demonstrated for like things like pH, where like the presence of soil and heterogeneity kind of provides some protection in the field for bacteria. And also, but that's something maybe Tom can also answer. So I know for the field, we basically Tau followed up the pH very well during the remediation, and that was really almost straight line. The pH was like in optimal range. I know by my own experience that in the lab, in a closed system, you can, there can be more. For example, if you add a bit too much electron donor, you can have like decrease in the pH. I don't know if Tom also followed up the pH.
Yeah, we did. Yeah, maybe I can add to that. So in the lab, what I mentioned already, we have a fully mixed system. So there is no way for the organisms to escape from the high chloroform concentrations. I think that's really an important factor. The pH during our experiments was in the optimal range. It declined a little bit, but not much. And of course, in the lab, we do a buffering of the bottles. Another thing is that the microbial community analysis, as we did it, is relatively unsensitive to the dechlorinating organisms. So typically we would have done, in addition to that, a qPCR analysis to just pick out the dechlorinators and see at which levels they are present. And here it is really, again, that COVID sucks because we just didn't have the chance to do it. There was no time in the lab to achieve that.
Yeah.
But that is what we normally do. So we look at the community composition and we look at some of the organisms that we are really interested in or that we think are important for the process that we look at. And our experience is that you get that qPCR, of course, it's more sensitive, but you get higher numbers in comparison to the community analysis. Also in a relative sense, because the community analysis is a relative abundance that you get out of that. But the 25% has not been there in the lab experiment. Let me be very clear about that.
Yeah.
It's just that we would have seen. And then maybe we would have seen 10 to 15%, but not the small amounts that we have. And I think that is really to the difference between the field situation and the lab. But when you think of that in terms of how relevant are then the lab experiments to the field situations, I think they are still relevant because also in the field you have the situations where organisms see these high concentrations in certain niches. And I also think you just have to be aware of these differences. So in the interpretation of the results, you always must be aware that there's a scaling effect between lab work, pilot tests, field work. And yeah, you have to take that into account when you interpret the results. But on the other hand, you can just learn from your lab experiments from the field. Yeah. I guess, but John has to confirm that. That's one of the reasons that GreenSoil is doing these lab studies.
Perfect. Yeah. I think that's about it for the questions. I don't know if anybody else has something to add. But if not, John, Tom, I really want to thank you guys for this presentation. I think it was a quite technical one, but a very interesting one. I at first quite learned some things, so I want to thank you guys for that. To the other participants, if you are interested in the slide deck of this presentation, just send us an email and we'll be glad to send it out. We will always also send out the recording via email to all participants. So should you want to listen to it again or forward to some of your colleagues, you will get it in your inbox soon. I hope we have provided some valuable content and insights. We are always available to hear from you and support you with our services. So I want to thank everybody also for joining the webinar. And as this was the last webinar of the year, a warm and good holiday time for everybody. We hope to see you all again in 2022, where we will be moving forward with these webinars, but where we also hope to be able to meet some people face to face, as Tom said, and I cannot state it in a better way. COVID sucks, so let's see what 2022 brings us. But once again, thanks for presenting, John, Tom, and thanks for joining everybody. See you all soon.
Thanks to the audience.
Yeah, thanks.
And thanks to you at GreenSoil for the invitation again.
You're welcome.
Tom also.
Take care, guys.